Welcome back to Cellular Chemistry Part 2. Today we're going to be talking about proteins and enzymes. So make sure you have your notes available and let's delve in. All right, so now we're going to start talking about proteins. So all six essential elements can be used in proteins. The subunit is the amino acid, which is made up of an amine group and a carboxylic acid for all those chemists out there. There are 20 different amino acids. Each one has the basic chain and then has a different specific side chain. Okay, amino acids come together and they create long chains, and the long chains are called proteins. All right, these proteins then fold into different shapes, and the shape of the protein will determine the protein's function. If the protein denatures or loses its shape, it can no longer function. So let's look at a particular protein. Let's look at hemoglobin. Hemoglobin is found in your red blood cells. It is shaped to hold oxygen. It's actually made up of 4 different proteins that come together to make this shape, and in the middle is an iron group that can work with oxygen. By bonding to the oxygen, it can carry oxygen throughout your body. Hemoglobin is super cool. It's a pretty large structure when you look at it from a chemical, from like the single chain, but it serves a very important function. There are a group of proteins called enzymes. Enzymes are shaped in a specific way to work with specific molecules. So proteins are the basic building molecules of life. They make up most everything that you are made of other than cells. Your hair is protein, your nails are protein. Proteins do lots of different things. They make up parts of your cell wall. They do a lot of different things throughout your body. We have proteins that are called pigments. There are proteins that absorb and reflect light. For example, chlorophyll absorbs light. It is a protein to gather energy for photosynthesis. It reflects the light green, and that's why it looks green. So the lights, just as a quick side note, the colors that you see on anything is what the light that is reflected. So it absorbs all other colors and reflects that color. So if something is blue, it is absorbing all colors but blue. White means it's reflecting all colors, and black means it's absorbing all colors. So some proteins are constructed to bind with foreign particles in the body, and these are called antibodies, and they fight illness. And some proteins form structures such as keratin, which is a protein that's in hair and nails. And we've got a lovely little picture of keratin here. So keratin's super cool. All right, and some proteins are used for transport through the cell membrane of the bloodstream, such as hemoglobin, and some are used for communication between cells. These hormones, for example, such as insulin, signals the use is a signal used in the use of glucose, and it is administered to treat diabetes, but your body naturally makes it. Other proteins serve as neurotransmitters such as serotonin or norepinephrine. Then again, that special class of proteins called enzymes. Enzymes work to speed up chemical reactions. So here's a lovely little video. The link will be provided on the class website. All right, so enzymes work to maintain homeostasis. All right, metabolism, or the accumulation of chemical reactions for energy use, requires certain conditions to occur. Enzymes regulate metabolism, allowing life to continue. Without enzymes, you would be dead. Enzymes speed up reactions by serving as a biological catalyst. Um, so in metabolism, each reaction has a small range of temperatures and pH at which it can proceed, okay? And every reaction needs a little bit of energy to get it started, all right? That energy to get it started is called the activation energy. Enzymes allow reactions to happen by lowering the activation energy. So let's look at a little graph. So a general reaction, if we're gonna look at this reaction, would take um, all this activation energy up to here, okay? So in order to start the reaction. When we add an enzyme, we lower the activation energy. It still requires activation energy, but it is much lower than what it was to start. And that allows the reaction to go to completion faster. See how long it takes for this reaction to occur compared to the catalyzed reaction? Okay, it's a lot faster. We need those reactions to go fast in the body so we can maintain homeostasis. All right, enzymes are usually proteins, and the proteins have a 3D structure based on how the chain folds. Okay, um, the enzyme has a specific place where the molecule that it is reacting with or working with can attach, and that special space is called the active site, and the molecule that is going to be attaching to that active site is called the substrate. Okay. If the active site shape changes too much, then the substrate won't fit and the reaction can occur with the enzyme. This means that the enzyme has been denatured. And things that can denature proteins include changes in temperature, pH, or salinity. And salinity is the salt content. All right, so then your enzyme won't work. So we have a range. If we have a lovely graph here that shows you the range for optimum temperature for an enzyme to work. Enzymes work at specific places best, and if they're not in that specific place, then they don't work. Lovely little picture of an enzyme-mediated pathway showing the enzyme with its open shape, okay, and how it has a specific space for the substrate to enter. Once the substrate enters it, it sort of closes in around it. You can't see that in this picture, but it usually closes in around it, does the reaction, and then out pops the products. Sometimes you'll have two things that will bond in the enzyme and one thing will come out. Sometimes it's one thing that goes in and two things come out, or sometimes even more. All right. They mediate the reaction using a specific chemical pathway. So they use a specific series of steps. It's not random. Okay. First, the enzyme collides with the substrate. All right. They fit together like a lock and a key. All right. The enzyme changes the substrate in some way, okay? It could either by break it apart through stressing the bonds, or it may hold two things together closely so that they can interact. All right, and then the enzyme and the substrate separate. The enzyme remains unchanged, just like corn, like I said in the in the biochemistry lesson. Okay, enzyme is unchanged by the reaction. So here's another little video that will be on the class. All right, enzymes have distinguishing characteristics. All right, they're specific, and they're specific because they have a specific shape of the active site. And they are reusable, okay? The enzyme does not change or split up, okay? That means it can repeat its role and be reused. All right, so I hope you have a great rest of the day, and I look forward to talking to you more about proteins and enzymes in class.